3 billion. That's the number of base pairs in the human genome. Now imagine trying to find one mutation that causes a disease manually. That's where bioinformatics comes in. Today we are exploring how this field didn't just help biotechnology, it completely revolutionized it. Bioinformatics is an interdisciplinary field that combines biology, computer science, statistics, and information technology to collect, store, analyze, and interpret biological data. Think of it as a data science of life. It transforms raw biological information from DNA sequences and protein structure to gene expression patterns into meaningful biological insights. Bioinformatics emerged as biological data began growing beyond the scientists could reasonably analyze by hand. And today, it has become a foundation of genomics, proteomics, system biology, and modern biotechnology. But what problems did it actually solve? Before bioinformatics, biotechnology faced a fundamental challenges. We could generate biological data faster than we could understand it. First, data overload. High throughput experiments can generate enormous amounts of genomic, transcriptomic, and proteomic data far beyond what human can analyze manually. Second, fragmented information. Biological information was distributed across scientific publications, laboratories, and emerging databases. Scientists needed computational systems to organize, compare, and retrieve this information efficiently. Third, limited predictive power. Without computational analysis, identifying genes, functions, comparing sequences, predicting protein structures, and connecting genetic variation to biological traits was far more difficult. And fourth, slow discovery cycles. In areas such as agriculture, identifying genes associated with valuable traits could require years of experiments and breeding. Bioinformatics helped turn these challenges into computationally tractable problems. But, its impact went far beyond solving existing problems. It opened entirely new areas of biotechnology. First, genomics and precision medicine. Bioinformatics made it possible to analyze entire genomes and identify genetic variations associated with disease traits and drug response. Second, drug discovery. Computational analysis can identify potential drug targets, predict molecular interactions, and support virtual screening, reducing the number of compounds that need to be tested experimentally. Third, modern plant breeding. Bioinformatics can help identify markers and genomic regions associated with traits such as yield, drought tolerance, and disease resistance. It also supports genomic selection, genomic analysis, and identification of targets for genome editing. Fourth, evolutionary biology and comparative genomics. By comparing genomes across species, scientists can investigate evolution, identify conserved genes, and understand how organisms have changed over time. Fifth, systems biology and multi-omics. Instead of studying one gene at a time, bioinformatics allows scientists to integrate genomics, transcriptomics, proteomics, and metabolomics to study biological systems as interconnected networks. And finally, synthetic biology and genome engineering. Computational analysis helps scientists design sequences, predict biological functions, identify suitable targets, and engineer biological systems with increasing precision. So, bioinformatics didn't simply give biotechnology better tools. It changed the scale of the question we can ask. But the next transformation may be even bigger. As bioinformatics continues to evolve, several major developments are emerging. First, artificial intelligence and machine learning. AI is increasingly being used to predict protein structure, analyze genomic variation, infer gene function, discovering drug candidates, and identify complex biological patterns. Second, pangenomics. Instead of representing a species with a single reference genome, researchers can analyze genetic diversity across many individuals and populations. This provides a much more complete picture of biological variation. Third, real-time genomic surveillance. Portable sequencing combined with computational analysis can help detect pathogens, monitor their evolution, and track outbreaks much faster than traditional approaches. Fourth, precision agriculture. As genomic information is combined with environmental systems and field data, crop improvement can happen increasingly tailored to specific environments and growing conditions. Fifth, increasingly powerful computational models. As computational methods continue to advance, scientists may be able to simulate biological processes at scales that are difficult to reach experimentally. And finally, there is a challenge that becomes more important as the technology becomes more powerful. How to manage, protect, and reasonably use biological data? Because the more we learn from genomes and more important privacy, data governance, accessibility, and responsible use become. Bioinformatics began as a way to manage biological data, but it became something much bigger. It changed how we read biology, how we understand it, how we discover new possibilities in biotechnology. And now, with artificial intelligence, multi-omics, and increasingly powerful computational models, bioinformatics is moving from analyzing biological systems to predicting them, and perhaps eventually designing them. The future of biotechnology may not be limited by how biological data we can generate. It may depend on how intelligently we can understand it. And that's the revolution bioinformatics has already begun.